Besides the HA and NA sequences, Librator also allows users to split any of their sequences into gene fragments. Users can use this function for some novel research subjects, for example, COVID-19 proteins and other sequences. Users can go to the Gibson clone and the Gibson clone for a single sequence to activate this function. So from here, users just need to choose your target sequence. For example, we choose Brisbane, and your sequence will be displayed here. And now there's just no joint region have been inserted. So here we have the input. You can just type your joint region 108 and click add. Then here you can browse your sequence, and this is the joint region you just added. And also here is overall the map that shows where your joint region is. Then you can add a multiple joint region, and all the information is here. Just 200, and this time we just type 201. So this one is too short. So for a Gibson clone, we set a minimal length for the joint region is 9 amino acids. So if you set it too short, Librator will notice you like your region is too short. Do you still want to continue? You can click yes. Then it will show the final product, the first joint region, and here is the second joint region. If you're not satisfied with current editing, you can just double click to remove this one. And if there are too many things to remove, then you can just click clear to remove everything. So here, let's just generate 200, 208, add 1, and the 400, 408, and another one. So now we added two joint regions to split your full-length HAs into full-length sequences into three different gene fragments. Then we can preview the final product here. So here are the 3 sequence tracks for fragment 1, 2, and 3. And we can just hide the nucleotide. Check the amino acid sequences, make sure they align well. After that, you should set up the upstream connector sequence and the downstream connector sequence according to the factor you use. Then you determine the output path for your files. Just go to send meeting, click open, then just click confirm. The results will be saved under that folder. We just open this folder. It's just a file. We can check the file. So it has all three fragments for your sequences, and all uppercase are your original sequences, and lowercase is the connectors. And beside that, actually this function also allows you to just design your full-length sequence into a 1 gene fragment. To do that, just choose your sequence, and without adding any joint region, just set up the upstream connector, downstream connector, and determine the output path. Just delete this first. Okay, just click open. Then you just click confirm. It will tell you no joint region determined will consider your entire sequence as 1 fragment. Click OK. And your results are generated. We just double check the results. So this time, Librator consider your full-length sequence as 1 fragment. And so here, the uppercase sequence is your original sequence, and a lowercase sequence is the connectors.